Hello. The topic for this video are going to be the organelles that are associated with eukaryotic cells. For this particular video, I'm going to focus on animal cells first. I'll have another shorter video where I'll kind of go over some of the organelles that are found just in plant cells. All right. And what I have over here, I've color-coded things for you. The list over here that's in blue and green, these are all going to be structures that are considered organelles. All of these are going to be surrounded by a cell membrane, so they're going to be surrounded by phospholipids and proteins. All right. On the other side here, I've got some other structures that are going to be important in the functioning of the cell, but these are not considered organelles. Okay, these are not surrounded by a cellular membrane. And I left one off here. We'll put this in yellow. We've also got the cytoplasm that we'll talk about a little bit about. Okay, so when I teach face-to-face, the way I like to do this is I like to draw a giant cell on the board. Ooh, that's a squeaky marker. All right, and label the different components. All right, now in terms of this class, I may expect you to be able to identify the various components that I'm identifying on this cell here, okay? I'm not going to ask you to identify anything that's not covered in the video or the textbook, all right? So we're going to start off with our eukaryotic cell here. So I've just drawn a big squiggly circle here. This squiggly circle represents our cell membrane, our plasma membrane. So that's composed of our phospholipids, our proteins, our carbohydrates, and our cholesterol. All right. That's going to contain the components of our eukaryotic cell. All right. And those components are going to be floating in a liquid. That liquid is the cytoplasm. Sometimes you'll see this referred to as kind of a jello or a matrix. So it's a little bit thicker than, say, water because it's got good stuff inside of it. There are going to be proteins that are floating around in the cytoplasm. There's going to be things like sugars floating around. There are going to be enzymes. There are going to be salts floating around in there. But they're all things that the cell needs in order to function, okay? So our cytoplasm is kind of going to be all of the negative space that I don't go over, all right? It's the liquidy stuff that's holding all the organelles kind of in place, all right? First organelle that I'm going to talk about is going to be the largest organelle inside of a eukaryotic cell. That large organelle is known as the nucleus. And I'll draw a little double strand here of DNA. This is going to be the function of the nucleus. This is going to be where the DNA is contained. Because with the eukaryotic cell, the DNA is extremely important. This is what dictates how the cell functions, how it runs. It's essentially the instruction manual for the running of the cell, okay? So it's important to kind of keep that all together. And our nucleus is going to be surrounded by a cell or plasma membrane. This is very often referred to as the nuclear membrane. And the nuclear membrane is actually a little bit different than our plasma membrane in that it has some pores associated with it. So I'm going to kind of take away some of my marker here. These are going to be our nuclear pores. So they're little openings. I'll draw a dot here. That's our nuclear pore. And what that allows is it allows for certain proteins in the cytoplasm, if they have a specific tag, to enter the nucleus, because the nucleus, based on its size, it doesn't want to bring things in that aren't supposed to be there. So if there are proteins that are needed for things like DNA replication, transcription, those things are going to be able to be imported into the nucleus, okay? There are also things that will go out of the nucleus through these nuclear pores, because when we talk about transcription translation, transcription is going to occur in the nucleus here, translation is going to occur out in the cytoplasm. So the RNA that's made for transcription moves out of the nuclear pore into the cytoplasm where it can be translated. And I know you don't know what transcription and translation are yet. Don't worry about that. What I want you to take away from this is there are things that can move in and out of the nucleus. That's why we've got nuclear pores associated with it, okay? All right, and associated with the nucleus then, all right, there are going to be some membranes that are going to come off of the nucleus. I'll draw those as kind of little wigglies, and I'm going to draw one up here because our membranes here that I'm going to talk about next kind of come in two different types. So this is going to be the endoplasmic reticulum that I've drawn here. And the endoplasmic reticulum can be either smooth or it can be rough. All right? And what makes the endoplasmic reticulum rough, I'm going to draw this as little dots. And what the little dots are here, these are ribosomes. Okay? And those ribosomes are going to assist in making proteins within the cell. All right? Ribosomes are found either attached to endoplasmic reticulum, in which case this would be the rough endoplasmic reticulum. The R, and I'm abbreviating this, the rough endoplasmic reticulum is going to be the one that has the ribosomes. Okay? This would be our smooth endoplasmic reticulum here. So this is our rough endoplasmic reticulum. This is going to be our smooth endoplasmic reticulum. All right? And the two different types of endoplasmic reticulum have different functions. Rough endoplasmic reticulum is primarily going to be involved in protein synthesis. Smooth endoplasmic reticulum primarily going to be involved in generating lipids, fats, and some steroids as well within the eukaryotic cell. So they both make something, but what they make is a little bit different. And I've drawn them here very separate. Just be aware that you can have endoplasmic reticulum that transitions between smooth and rough, so they may all be connected. Don't let that throw you, all right? If it has ribosomes on it, it's going to be rough endoplasmic reticulum is involved in protein synthesis. Smooth endoplasmic reticulum going to be involved in lipid hormone production and things like steroids. Okay. You will also find ribosomes, and now my cell is going to look very polka dotted, and when I stand in front of this, it's going to look like I've got acne. All right. They can also be out in the cytoplasm. All right. These are termed free ribosomes, not because they don't cost anything, but because they're away from any other organelles. Okay. The free ribosomes also involved in making proteins for the cell. All right. So that's our smooth and rough endoplasmic reticulum. The next one that I'm going to talk about is going to be our Golgi apparatus. And the Golgi apparatus kind of looks like a stack of flattened pancakes. And that Golgi apparatus, as far as its function within the cell, is going to be in packaging. So what's going to happen inside our cell? You're going to have proteins that are made in the rough endoplasmic reticulum. You're going to have parts of that membrane that are going to bud off, and they're going to migrate to the Golgi apparatus. The The Golgi apparatus then is going to take those proteins and it's going to package them and send them where they need to go within the cell. All right? So I kind of think of the Golgi apparatus a bit like the post office of a cell, because it sorts out what comes in, and it'll sort out things also that come from the smooth endoplasmic reticulum as well. Okay? So it'll receive packages from the endoplasmic reticulum, sorts it out, and then sends those where they need to go. One place that those proteins made within the endoplasmic reticulum, the rough endoplasmic reticulum, may need to go is going to be to our cell membrane, because we're going to need to replenish the cell membrane. You can't just keep the same cell membrane, because over time it'll kind of break down. So those bits of membrane that come off the endoplasmic reticulum and come off the Golgi apparatus renew the cell membrane. It can also release whatever product, I'll make a yellow dot there, yellow dot, okay, happens to move through our Golgi apparatus, okay? So the endoplasmic reticulum, the Golgi apparatus, and the last one I'll talk about, the lysosomes, are all part of the endomembrane system. And I'm going to draw this one kind of big. Relatively speaking, structurally it wouldn't necessarily be this big within a cell, but to kind of, goodness, I can't spell lysosome. Lysosome. There we go. To show you what's happening with the lysosome, I'm just going to draw it bigger. All right? The lysosome would be something that would come off of the Golgi apparatus. And what the lysosome will have inside of it are going to be digestive enzymes. So the lysosome is responsible for breaking down materials within the cell. So if it encounters something like an organelle that is out of commission or isn't functioning properly, the lysosome can engulf that non-working organelle and break it down. Okay? It can also break down if you've got stuff moving into the cell by endocytosis. If the cell's bringing in materials, it will break down those cellular materials, and then the cell can use the components of what's inside of that. Okay? So the lysosomes kind of work to digest. Oops. Goodness, my spelling is just horrible today. Digest and really recycle the components that are within those various vesicles, all right? And a vesicle really is just another term for describing something small that's surrounded by a cell membrane, okay? Last organelle that we've got, I'm going to draw in a different color because I'm going to talk more about this one, because this is going to be an important organelle. And that's going to be the mitochondria. All right, that mitochondria, the function of the mitochondria, this is going to be the power plant within the cell. This is going to be where cellular respiration happens. So we're going to talk a lot more about the mitochondria in some upcoming videos. All right, mitochondria, I still did not spell that right. Goodness, take off points for spelling from Dr. Riley. Mitochondria, that's better, all right? It's going to be the powerhouse. This is going to be where cellular respiration happens. This is going to be where ATP is generated within the cell. So this is a very important organelle, all right? So that kind of covers the bulk of our membrane-bound organelles, all right? Some of the other structures I haven't talked about yet are the nucleolus and the cytoskeleton. The nucleolus is a darkened area that's found within the nucleus. And this is going to be one area where you want to pay close attention to because the spelling between nucleus and nucleolus is very close. I will take off spelling if you put nucleus and mean nucleolus, all right? And I want you to know the difference between what they do. So the nucleolus is found kind of as a darkened area in the center of the nucleus. This may be another structure that you can visualize using a light microscope if you're taking the lab. So our nucleolus here, this is going to be where ribosomes are going to be made within the cell. So the ribosomes that are out in the cytoplasm are actually synthesized or made inside the nucleus. That's going to be an example of something else that's exported out of the nucleus. So those ribosomes move from the nucleus out to where they function in the cytoplasm. Okay? Hopefully this made some sense. If not, ask me questions. I've got another video coming up. I'm going to discuss the cytoskeleton separately from this because this diagram now has gotten very busy. So we'll have a separate video on the cytoskeleton, and I'll have a short video on the plant showing some of the structural differences between an animal cell and a plant cell. Thank you for your attention. If you have any questions, please let me know.